So thank you, Kathy, very much for that very generous introduction. Charlie Nemiroff, thanks for bringing me here. It's always great to visit Miami, and my parents lived in Broward County for 25 years, so I have a long history of coming down here. I'm sorry that I brought this northeastern weather with me, although it's still a lot warmer here than it is when I go back home tomorrow. So I'm really very delighted. I'm very touched by this crowd because it's really quite impressive, and I'm very delighted to have an opportunity to talk to NAMI people, members, people who have an interest in the NAMI organization. I've been related in various ways to NAMI for 30 years. I've been very involved in NAMI conventions. In fact, I remember vividly, I don't know if anybody else in this room, maybe Charlie, of the first NAMI convention, which was in New Orleans. And I think it was in about 1980 or 82 was the first NAMI convention. I remember it vividly because it was in July. NAMI had no money. And this convention, which was the first annual meeting of the National Alliance for Mentally Ill, was held at Tulane University in some conference room. And all the people attending were sleeping. This was in July in New Orleans, which is very hot. And everybody was sleeping in the dormitory on the mattresses without sheets. Those were college student mattresses, which were all vinyl, no sheets. And what I remember was I slid off that bed in the middle of the night, landed on the floor because it was so hot that if you sweat, if it's a vinyl mattress, you just slid off the thing. So that was my first introduction to NAMI, and NAMI is an extremely important organization that is very dedicated towards helping families deal with the incredible difficulties of having a family member with serious mental illness. And this is something that, as Dr. Nemiroff mentioned, he certainly has been at the forefront of trying to understand the causes of these conditions to try to improve the lives of people who suffer with them. And I'll tell you a little bit about, this is going to be a science talk, but I'm going to try to make this understandable about where the science is to try to move to a new level of understanding of, I'm going to focus on schizophrenia, but this is really, to the most part, true of bipolar disorder. It's true of serious mental conditions, to try to understand how the new science can help us improve the lives of individuals that suffer from these conditions. So if I can have the next slide. The first thing is just to remind you that mental illness are very common disorders, and they're strongly genetic. I love this quote of Cary Grant, "Insanity runs in my family, it practically gallops through it." We've known for about a century, maybe longer, that mental illnesses run in families. But what we discovered in the last 60 or 70 years is that the reason they run in families is not the reason that language or religion runs in families. Language and religion runs in families because it's a cultural aspect of the family. It's something you learn in your family from your parents. Mental illnesses run in families because of genes you inherit from your parents. And that's the reason they run in families. And this was determined by very detailed scientific studies of conditions like twins who are separated at birth, where they don't share any of the same childhood experience, but they also have a much greater likelihood, if one has a serious mental illness like schizophrenia, bipolar disorder, the other does also. About a 50% probability. There have also been many studies where children, usually in Europe, were adopted away at birth from a parent who had a serious mental illness. And when you're adopted away at birth, you have no life experience of the ill parent. And what they showed in these adoption studies, that if you were adopted away from a parent who had a serious mental illness, even if you were raised by parents with no mental illness, you had a much greater likelihood of manifesting the same illness as your parents had than other adopted away children whose parents did not have a serious mental illness. So these were the kinds of studies that established that what was creating the risk probability in a given individual was something they inherited, not something they experienced. These were very important studies because many of you may remember, and when Dr. Nemiroff and I trained in the 1970s and 80s, we were told, as many people who studied schizophrenia and bipolar were told, that early childhood environments had a lot to do with the causes of these illnesses. We now know that early childhood parenting has no role in the causes of mental illness. The behavior of mothers who were blamed for many of these conditions bears no relationship to the causes of serious mental illness. Who is at risk is determined by genes. Next slide. So we know that next slide. Hello? Sorry. Okay, so let me just give you a sense of why we get very excited about genes. Because genes are the first clues that we have ever had to the causes of mental illness at a very, very basic level. Genes are the game changer in psychiatry. And the reason they're the game changer is prior to discovery of genes which change somebody's risk for mental illness, all we knew about mental illness was what it looked like, what it sounded like, to some degree what it felt like, but we really did not know what it was. Now, genes are not the causes of mental illness by themselves. Genes are they're risk factors. So genes for mental illness are not genes for fate. Genes for certain genetic disorders like cystic fibrosis or Huntington's disease are genes for fate. If you have those genes, you have the illness. Genes for mental illness are like cholesterol. They're genes for risk. They don't give you illness, they change how at risk you are. So if you have an elevated cholesterol, it doesn't guarantee you a heart attack or a stroke. And most people with elevated cholesterol never have a heart attack or a stroke. But it increases the chances that if you accumulate other risk factors, like high blood pressure or obesity or other things that are also risk factors, and you also have elevated cholesterol, your risk is that much greater. So we know that the genes for mental illness are like genes for all common human disorders. They're like the genes for diabetes, they're like the genes for cancer, they're like the genes for asthma, they're like the genes for heart disease. These are genes that change how at risk you are, not whether you have illness. So genetic insights are by definition clues to causative mechanisms. And because they're by definition clues to causative mechanisms, they have to be clues to treatments based on causation and not on appearance or features of illness. I like to say that in psychiatry, there are basically three treatments, three classes of treatments in the whole field. There's talking therapy, there's medical therapy, and there are electricity therapies. Those are the three therapies, which in many, many people are very effective. They're not as effective as we want them to be. They all have untoward effects that we wish they didn't have. But in many people, they're very effective. They're certainly more effective in treatment than people's lives were before they were discovered. But none of these treatments, talking therapy, medical therapy, electricity therapy, none of these treatments were discovered with any understanding of the causes or mechanisms of mental illness. They were all discovered in one way or another by accident. Just happened to make somebody a little better. The classic story of lithium, which is probably the most effective treatment in psychiatry for people with a particular form of bipolar disorder, was discovered by accident in Australia as a salt substitute. And a general medical officer made the observation that when people were taking lithium chloride as a salt substitute because they had high blood pressure and they shouldn't be on sodium, if they happened to have bipolar disorder, they seemed much better. That was the discovery. Chlorpromazine, which was the first antipsychotic drug, that was the forerunner of Haldol and clozapine, all these drugs, was originally tried by a French neurosurgeon as an antihistamine, post-surgically. And he said to one of his psychiatric colleagues, I gave this drug to a patient of yours who had surgery to dry out his bronchial tubes as an and he was much better. His psychosis was much better. This is how these drugs were discovered. So now that we have clues to what some causative mechanisms may be, this has to be a better way to find treatments. And it has to be a better way to find treatments that will not have the same kind of unexpected, undesirable side effects. Genetic insights also clarify the environment. This is a very important point. because we know the environment's important, but we don't understand the environment. It's very easy to say, oh, stress is a bad thing. Of course it's a bad thing. But we also know that every individual has a different way of experiencing stress. Some people experience stress as a challenge, as an opportunity. I always like to say, when there were 5 minutes left in the basketball game, Magic Johnson, certain people wanted that ball, and they didn't want anybody else to have that ball. And they saw those 5 seconds in the last shot of the game as the greatest opportunity. Other people would be terrified to have the ball at those 5 seconds. So environment is very personal. And clearly, can you go back? We got ahead of my, okay. Clearly, the reason environments have different effects on different people is because our genes are different. We're all different. So if I look at this room, nobody in this room looks exactly like anybody else in this room. I don't think there are any identical twins in this room, but we all look different. And it's not just our faces that are different, our brains are different. We respond to experience differently. We respond to stress differently. We respond to disappointment differently. We respond to challenge differently. Human beings are an incredibly diverse species. And this genetic diversity is critical for how we've survived all these millennia, because human beings would never have made it through all the droughts, all the infections, all the poisonous plants that people were eating 30,000 years ago when they didn't know what was a healthy plant from a poisonous plant. The only reason we survived all these challenges to human existence was because there was always some people that could survive anything. There are people who are immune to AIDS. There are people with genetic variations in their genomes that make them immune to AIDS. And there were always people that were immune to the latest virus or the latest poison in the plants that everybody was eating. So some human beings have survived all these incredible historical challenges to human survival because of this incredible diversity. So in order to understand how the environment matters to an individual, we have to understand their genomes. We have to know what is in their DNA that makes them different. And that's the principle of how we can uncover the mechanisms that will be individualized, where the holy grail now is this concept of personalized medicine based on individuals because we understand their unique features. Next slide. So let me talk a little bit about schizophrenia. And so there are 3 things we know about what puts somebody at increased risk of schizophrenia. The first thing we know is schizophrenia is like every common medical disorder. And by the way, I'm going to talk about schizophrenia, but this is true of autism, this is true of ADHD, this is true of bipolar disorder, this is true of most major psychiatric illnesses. There's no gene for mental illness. Genes do not know about a hallucination or a delusion or a panic attack. Genes know about how you build a brain. Genes know about how you make a cell work. So genes are about 2 things. They're about these fixed characteristics that we have all of our lives. How tall we are, what color our eyes are, what color our hair is. These are things, the fixed traits that are determined by genes, heredity. The other thing genes do is genes are the blueprint that every cell in your body uses every second of its life to tell it how to live. Because genes, which is in this DNA, is the blueprint for building proteins. And the proteins are built by assembling strings of amino acids in a cell, and the recipe for doing that is in the DNA. So how a cell makes an enzyme, how a cell makes a protein that gives it a membrane that allows it to be a 3-dimensional structure with integrity, how a cell moves, how a cell builds connections to other cells, which cells in the brain do. This is all determined by the construction of proteins that are based on your genetic code. So genes are about fixed traits you have all of your life that you inherit from your parents, but they're also about the biology of how a cell works. So we've known, if you could push the next slide. First thing we've known is that these disorders are what's called polygenic. There are many, many genes across our genome. When I use the word genome, I mean all the genes in your DNA. So in a human being, there's about 20,000 genes that are dictated by the 3 billion letters of our DNA code. The DNA code has 3 billion letters in it, and that makes about 20,000 genes. That's a little bit misleading because those 20,000 genes can be assembled in various ways to produce several million proteins. Every cell has the capacity to make several million proteins out of those 20,000 cells. And that's how the cell manages to survive for 70, 80, 90, 100 years in some people. So we've known for a long time that there's no one gene that gives anybody a serious mental illness. There are combinations of many genes, just like there's no one gene that causes a heart attack. There's no one gene that causes the common form of diabetes. There's no one gene that causes asthma. These are caused by many different risk factors that combine to create a state of risk. That's the first thing we know. The second thing we know, next slide. The second thing we know is that early life environment matters. When I say early life environment, I mean intrauterine life environment. And there's a number of pieces of evidence that have suggested that what happens during the period of time when brains are being built, which is during prenatal life, when brains are being built, is very important for the expression of mental illness, even during adulthood. So how do we know this? So in the world of schizophrenia, this is true of other conditions also. Certainly true of autism, ADHD, dyslexia, Tourette's syndrome, and to a lesser extent bipolar disorder. If a mother is pregnant during an influenza epidemic, or is pregnant during a time where there is a major famine, this has been studied around the world, there's about a 2-fold likelihood that an offspring from that pregnancy will have one of these developmental behavior disorders. It increases the likelihood about twice what it would be otherwise. And there are many, many other conditions that affect the pregnancy. If you don't gain enough weight because of malnutrition, or you gain too much weight, it increases the probability. So many things that can affect what we call the intrauterine environment that nurtures the development of a fetus can affect how a brain develops that ultimately puts somebody at risk to manifesting these conditions later in life. This is what we call the developmental trajectory. The developmental trajectory, if you want to think about how this works, I like to use this metaphor of bowling. When you go bowling, you want to roll this ball, and if you want to hit a strike, the goal is to make that ball go right over that first arrow that's just to the right of the midline on the bowling alley, about 10 feet from the starting place. If you get the ball to roll right over that arrow and has a little bit of a twist, it's going to land in the strike zone. If the ball is 1 centimeter or a ½ inch off that arrow, by the time it gets to the pins, it's not a ½ inch off the pins, it's about 3 or 4 inches off the pins, because it gets exaggerated as it moves down the alley. That's development. The destiny of that ball is determined based on its relationship to that arrow when it first hits that part of the alley. And as it moves down the alley, which is the period of development, this effect gets amplified. If it's 3 centimeters off the arrow, it's going to be way in the 7 pin. If it's 10 centimeters off the alley, it's going to be in the gutter. So this is a way of metaphorically thinking about how getting the trajectory right from the very beginning has long-term implications. We like to think that this is the genetics. The genetics is how much you are right on that arrow or not. What happens as the ball moves down the alley is that there is dust on that alley, which is environmental experience. And the dust can deviate the trajectory further by pushing the ball further to the right, or it actually could push it further to the left and rescue some of the genetic predisposition. Where the ball goes relative to that arrow is the genetic predisposition. What happens to it as it moves down the alley is the environmental experience that follows that developmental, early developmental phase. Part of the reason that we know that what happens in intrauterine life matters to your risk comes from studies of fraternal twins and studies of siblings from unrelated pregnancies. Identical twins share all of their genes. They are identical genetically. Fraternal twins are really just like siblings. They come from the same pregnancy, but they're completely separate genetically. And they're like any pair of siblings. They share 50% of their genes. All siblings are 50% identical, 50% different. Identical twins are 100% identical genetically. Siblings are 50% genetically identical. But if you look at the probability that if one member of the sibship has schizophrenia, what's the probability that the other has it? So if you're siblings from unrelated pregnancies, if one sibling has schizophrenia, there's about a 1 in 10 chance that the other sibling will have it. But if you're dizygotic twins, meaning you're the same genetically alike as siblings, but you share the same intrauterine environment, there's a twice greater probability than the average sibship that you would have schizophrenia. And this suggests that something about intrauterine life matters to risk for manifesting these conditions later in life, independent of genetical identity. Next slide. The last thing that we've known for a long time, which we have very little understanding of, is that all developmental behavior disorders, schizophrenia, autism, ADHD, dyslexia, Tourette syndrome, all of these conditions are 2 to 4 times more likely in males than females. And there's been no understanding of this at all. So now I want to tell you some of the recent research data that suggests a way of maybe understanding these phenomena. Next slide. So this is the current state of the genetics of schizophrenia. And I don't expect you to process this, but this is just to make the point of where we are in finding genes. So the whole landscape has changed because of the DNA technology revolution in medicine. We now can we can now measure, we can count everybody's DNA code. We can measure it, we can determine it. It's relatively inexpensive. When I started this in the late 80s, early 90s, finding genes was like a needle in a haystack. I like to say today it's high school biology. In high school biology classes, people do gene discovery because it's become very, very relatively simple biological research to find genes and to be able to study DNA. So we've known in the upper left-hand corner, this is just to show you if you have one member of your family with schizophrenia, what is the probability that you in the same family will have schizophrenia? So if you're one of identical twins, it's 50-50. And depending on how much you are related to the person who has illness, your probability drops off pretty quickly. So if the general population is a 1% chance that anybody has schizophrenia, if you're a first cousin, you've got a 2% chance. There's a twofold increase compared to the general population, but still very small. If you're a fraternal twin, it's 70%. If you're a child, it's 13%, et cetera. So this was the data that showed that these things ran in families. But now that we can measure genes directly by doing DNA analysis, there have been 3 different ways people have found genes for schizophrenia. The first is to actually read every single letter in the DNA alphabet in the parts of the genome that make proteins. And what's been found is about 1 out of every 200 people with the diagnosis of schizophrenia has what's called a mutation. They have a rare change in a DNA letter that affects the amino acids in a protein and affects the function of the protein. And about 1 out of every 200 people that we diagnose with schizophrenia, which is rare, an infrequent thing, has a mutation that likely is a major factor in the fact that they're ill. And because that mutation is making a particular protein not work right. There's another 2% of cases that have a larger change in the DNA code involving either the loss of segments of DNA. These are called copy number variations, or CNVs. About 2% of cases have one of these things. About 1 out of every 50 people have one of these. These are, again, they don't necessarily account for schizophrenia in those individuals, but they're likely to be important risk factors. 97% of people that get the diagnosis of schizophrenia have none of those. They have combinations of the common variations in the human genetic code that all of us have some of. So all of us have some of the genes that have been linked to schizophrenia. People that have the diagnosis have a bigger burden of them, which is why they're at increased risk. Everybody has a few of these, just like everybody has some gene for some medical illness that increases the chances of their medical illness. We're all at different risk states. Some people maybe have a risk state of 90%, some people have a risk state of 5%. We all have some risk state for these conditions. And the way these are discovered is by these studies which are called GWAS studies. And these are studies where you just basically assay every variation in the DNA code that differs between people, that accounts for everybody being different, and looks at whether any of these are more likely to be found in people with a diagnosis of schizophrenia or bipolar disorder. When you find that, you say this is a region of the human genome where a gene for schizophrenia is likely found. So that's the state of the art. Next slide. So we've learned a lot of lessons from these studies. These are the take-home lessons. The first is there is no gene for psychiatric illness. Very important to realize this. Just like there's no gene for heart disease, there's no gene for stroke. There are genes that increase the risk of any of these common disorders. The genes they're not genes for fate. They're genes for how much any individual is at risk. And obviously, if you accumulate other risk factors, that risk accumulates. There is no royal road. There's no one way you get there. There are many different roads from a genetic point of view, many different ways you can inherit different combinations of different variations across these 3 billion letters that give you risk. Nobody is at risk because of one gene or another gene. You're at risk because of the whole complement of all the genes in your DNA code, which is referred to as the genome. The genome is all the genes that you have. And the environment has to matter. We know this. But again, the environment is complicated. Everybody, when you think environment, you think, oh, did you have an argument with your father? Did you have a difficulty at school? Did your girlfriend reject you? You know, we all have ideas what the environment is. But it's very important to remember that for a cell, which is what we are made of and what DNA is about, DNA is only about cells. The environment of a cell is the molecules that are floating around outside the cell that are talking to the cell. Cells only know about biochemistry and biology. They don't know about mothers or fathers or schoolmates or anything else. So in theory, whenever one has an experience that might be stressful or that might cause someone to feel disappointed or angry or upset, there are molecular changes that are taking place that have impact on a cell, and the DNA is responding to this. So these are the lessons that we learned. How can we try to make some sense out of this from the point of view of mechanisms? Next slide. Okay, so this is the schema for thinking about how genes translate into the mental conditions that are so disabling and that are the reason that organizations like NAMI exist. So genes, this is sort of the landscape of how genes relate to behavior. Genes are about cells. Genes only know about the biology of cells. Genes only know about making proteins so a cell can do what a cell has to do. Genes don't know anything about cognition. They don't know anything about perception, thoughts, mood, etc. Cells handle molecular information. That's what cells do. The cells work together to build organs. Genes don't even know about organs. They know about cells. But cells become tissues. They become organs early in development. So each of these levels of analysis involves an increasingly complicated kind of biology. Cells process molecular information, molecules. Tissues process more complicated information. So cells in the brain become systems of the brain that process environmental information. Brains process sensory information, they process emotional information, they process cognitive information. Genes know about cells, which process molecular information. Cells form tissues like a brain, which processes environmental information. And what we call behavior is presumably how these multiple levels of increasingly complicated biological function are observed by us in our complex social environments, that we see that the outcome of all of this. So to understand how a gene, through this big red arrow, ultimately has anything to do with behavior, we have to understand how it affects these cells and how the effects it has on these cells affects the building of a brain and the tissue and the circuits that these cells are important for. Ultimately, psychiatric illness is about how the brain processes information in the environment. That's how we recognize it. Let me give you a metaphor, which I'm very fond of. Next slide. This is like thinking about how you go from the musical score that Beethoven wrote for Beethoven's Fifth Symphony to something that we hear as Beethoven's Fifth Symphony. Because if you think about how this happens, you have all these little instruments, which are the cells. They each have this DNA code, which is the musical score, that they read. This musical score has absolutely no meaning whatsoever, except that the cells know what to do with it, which is the violins or the brass or the percussion or the strings. Because all this is, these are just contrasts on a strange thing. They're just nothing, except they have meaning to these instruments. And these instruments read that and do something with it. And these instruments become part of systems in the orchestra. The strings, each violin has to be in tune with the other violins to be the strings. And they have to be the brass, and they have to be the percussion. And together, these sections, which are like the neural circuits of a brain, each processing different components of what this score, which is the DNA code, is telling these instruments to do. Ultimately, if all of this is done the way it's intended, we recognize it as Beethoven's Fifth Symphony. But if the violins are out of tune, and they're not in phase with the other instruments in the string section, the string section is no longer in tune with the brass and the percussion and the woodwinds. And what comes out is not Beethoven's Fifth Symphony. So this is in many ways, I think, a useful metaphor for thinking about how, starting with these genes, we end up with very disordered brain function, very disordered representation of what's meant to be a particular organized pattern of behavior. Here, Beethoven's Fifth Symphony. Next slide. So this is the challenge of our time. And I come representing this Lieber Institute for Brain Development, which I'll tell you about at the very end, where the challenge that we've been faced with is say, let's take this way of thinking about how genes affect the biology of cells and the development of brain systems that mediate complex environmental stimuli, and use this information to somehow come up with treatments based on an understanding of mechanisms and not based on coincidence, accident, or just pure happenstance. This is really the challenge of our time, I think, is to use this new information that we've acquired about how what basic biology of mental illness might be, to come up with new strategies for improving the lives of affected individuals. Next slide. So let me just talk. I'm going to spend the next 5 minutes, I'm almost finished, talking about what we think the nature of this inborn genetic bias, and starting that bowling ball down the alley, what it really means in terms of how this affects early brain development. So we know, anybody that has children knows, that there are certain aspects of human temperament that are there from day one. There have been many studies of this. Jerome Kagan was a very well-known psychologist at Harvard University. He studied children the first 2 weeks of life and showed that many of the character, and I think you all know this as parents, many of the characteristics of your children in the first weeks of life, in terms of their sensitivity to environment, how they respond to discomfort, how they respond to being nurtured or supported or coddled, how they respond to colic. Many of these temperamental characteristics are there from very early in life, and they remain relatively consistent features. Obviously, they have different, it's not colic when you're 20, it's different features of environmental stress. But these features are born in very early in life, likely in the genetic programs. And so we were very interested in the possibility, next slide, that there are early developmental components to mental illness. That they, way before anybody diagnoses these conditions, there might be aspects of how the bowling ball is offline from very early, but at a stage where you have no idea that by the time it gets to the pins, it's not going to be a strike. You wouldn't know that from where the bowling ball is at that time. Next slide. So we've done a number of studies, and I've been interested in this for over 30 years. And in fact, 31 years ago, this month, I published a paper in the American Journal of Psychiatry suggesting that if we wanted to understand schizophrenia, maybe we should stop focusing on the time that the diagnosis is made and start focusing on how brains get built and what sets them on this trajectory from very early in life. And maybe if we had a better understanding of this, we might have a better understanding of what these conditions are. And I also went so far at that time to suggest that these may not really be illnesses in the traditional medical sense of the word, but they're states of brain development that have a particular way of interacting with personal experience over the lifetime. They are this trajectory down the bowling alley rather than some illness in a typical medical sense where these cells are dying or they're sick or something else. So there have been many, many studies now, next slide, of people with schizophrenia. These are mostly done in Europe, where there are these big population-based public health registries where you can look at every person ever hospitalized in a country like Sweden or Denmark or England, and you have all this early life history data on them. And many of these studies were done showing that people who are diagnosed with schizophrenia as adults have subtle variations in development from very early in life. So for example, it's been shown in Finland, Denmark, Sweden, and England that as a population, the people who are diagnosed in their 20s with the diagnosis of schizophrenia sit up on average, not any individual, but the whole population. The average human being sits up at 26 weeks of age. That's the average. Some human beings sit up at 24 weeks, some sit up at 25 weeks, some sit up at 27, 28. But the average, right in the middle, 26 weeks. The average person who is diagnosed with schizophrenia in their 20s